Hello, I'm Teemu Daniel Laajala, and welcome to our poster presentation titled A Harmonized Resource of Integrated Prostate Cancer Clinical, Omic and Signature Features. You can find myself and my colleagues over here, as well as our affiliations and funders over here. It's a published work, so if you want to take a look at it, it's in Scientific Data, and I've also put great effort into putting everything as open source, so here's a link to our GitHub. The motivation behind the work is that there are a lot of open prostate cancer datasets out there. However, what we found was a lack of effort to try and harmonize those studies so that they could be leveraged together. So, we identified 19 datasets and tried to process them in such a manner that they would be comparable to each other. We ended up with over 2,900 primary tumor samples, over 200 normal tissue samples, and over 500 metastatic prostate cancer samples across these 19 datasets. And all of these are offered via an R package called Curated PCA Data. The key ingredients behind the R package is that we're pulling our data from cBioportal, Gene Expression Omnibus, and Xena Browser, the last one primarily for TCGA. And what we do for you is we try to scrape and extract all the potential metadata that is interesting and harmonize that to the same level, comparable to each other. And we go to the rawest possible data level. For example, for microarrays, we go to the level of cell files. And there we use the latest gene annotations and harmonize the pipeline for processing those files. For example, in the sense that for most microarray studies you'll find that we used RMA normalization, making them better comparable to each other. I mentioned it's an R package and it's already been accepted to Bioconductor. As such, it's mainly composing of classes that are Bioconductor friendly, so the studies come as multi-assay experiments, which are downloaded from the cloud service experiment hub. And inside these objects you'll find standardized metadata tables, where you will find key information such as patient's age at diagnosis, PSA at baseline level, and for example Gleason grade. And we chose to focus on 3 omics levels: gene expression, copy number alteration, and somatic mutations. And mainly from the transcriptome, we derived new variables via multiple methods of immune deconvolution, and we also calculated risk and AR scores. And all of this comes in R with a lot of user friendliness via accessor functions and convenience functions, as well as vignettes which showcase the work. In Table 1, you can find a summary of the sample counts in respect to different strata, for example the sample types. You can for example notice here that Abida et al. Is a study focusing mainly on the metastatic samples. But for most of the data sets we find primary tumor samples complemented by normal samples, for example in Taylor et al. And TCGA. Most of the omics do focus on transcriptomics, as you can see from this row over here, but we tried to identify as many as possible multi-omics datasets, and if you look at the intersection of the multi-omics, we are able to also provide datasets where you have all three levels of omics available. And we also provide relevant clinical endpoints, for example biochemical recurrence, overall survival, and Gleason or grade group, if these just were available. Taken together, we demonstrate in the paper how you can use these datasets together and find interesting things and consistency in them. In Figure 1, we, for example, identified that elevated endothelial cell counts estimated with 3 different computational methods, EPIC, MCP counter, and Excel, were acting consistently in TCGA and Taylor et al. In the sense that elevated endothelial cell counts, the top 1/3, had a higher risk of BCR in comparison to the bottom 2/3, with a statistical significance in the Kaplan-Meier curves. Then, showcasing Taylor et al., TCGA, and Baca et al., we picked some key genes over here. For example, the ERG-Temporus2 fusion. You can notice that that was having a gene fusion at roughly 40 to 50% of the samples. And if we look at oncogenes, for example, the MYC over here, we notice in Taylor et al., TCGA, and Baca et al., MYC was amplified consistently. And in contrast, P10 and TP53 were deleted. And we've already utilized this R package in, for example, Gilson et al., as well as developing new computational methodology in Halkola et al. So hopefully you'll find this data resource useful, and I'll be happy to answer questions or talk to you hopefully in person. Thank you.